second discussion session for this particular course, Neurobiology, that you guys have enrolled in. And I'll just be sharing my screen and we can begin with our discussion pertaining to this session now. Yeah. So the format that we follow will be similar to what we have been for all of these discussion sessions pertaining to the NPTEL courses that I have been taking, right? Just a moment. I'll just share my screen again. In case there is any lag, please let me know. I'm on a different network connection today. So, for those of you who are new, we have already gone through one round of this kind of a live discussion session for this particular course, that is NOC 5 BT77 or neurobiology. And over there, we had discussed, the techniques related to the brain imaging and disease pathology identification. Those things we were discussing. We have not done it we have not completed that discussion. We'll take it after we are done with week two's assignment discussion, right? So we'll come to that as well. For now, people who are new, I would like to introduce myself. I'm Arveen. I'm a PhD scholar at Department of Biological Sciences and Bioengineering at IIT Kanpur. And as a PhD student in my first year, I too have studied this course, neurobiology, which was taught to us by Professor Nitin Gupta. Over here, you have his recorded lectures from NPTEL that are pretty much similar to what we had been taught offline as well. And in case you are aspiring to learn a little bit about neurobiology or to, like if you are just curious, you want to learn, it is a very basic level course. It will really help you. His lectures are fabulous. There are books that he has mentioned, such as Candle, which you can refer to, which again is a simply written book. You will understand most of it if you have a good grasp in English. It is not too difficult to read. And apart from that, there are some concepts that, because of the short duration of the course, might not have been taken up in his lectures itself. So that part we'll be emphasizing more on in these, live discussion sessions. Also, if you have any doubt, you can ask me. We will discuss the assignments pertaining to that particular week's lecture. And along with that, what we will also do is once we are done with, discussing about anything new that we want to discuss, we will also kind of revise a little bit or we will go through the overview of what has been taught to you in that particular week. Right. So this is more or less that kind of a session. Yeah. So what we'll do is, without wasting any further time, we'll begin with week two's assignment first, because we have a part of week one discussion also still left that we want to complete today. Yeah. So how we'll do this is that I have first just given you the question and the options pertaining to those questions. We will go through this and then we will determine, then we'll come to another slide which has the answer for this question and also the reasoning behind that or the concept behind that answer. Right? So we'll be discussing in such a manner. In case, I would like to encourage you to answer alongside, so that if there is any doubt, we can clear it right away, right? If not, I'll still go through with the reasoning, with the concept explanation. So I hope that also helps. Yeah. So in that case, the first question for this week was, if a small positive current is passed into a neuron, the voltage of the neuron relative to outside will increase. Is this statement true or false? Right? So for this, you should have an understanding about if a positive current is being passed, what will be the effect of voltage in general? What is the resting voltage of a neuron in general? And so that you can account for what kind of effect this small positive current will have on that resting potential, right? Will it increase? Will it decrease? What will happen, right? So the answer for this one, it is a simple true false question. The answer for this one is true, right? So that means that if we are introducing a small positive current into a neuron, right? So means positive charges being sent into the neuron, right? So the voltage will increase. Yes, it will increase. Generally, what is the voltage? It is either minus 70 in our case or minus 65. We were studying in the case of squid giant axon, right? So it will increase from that, it will come to less negative, right? That is also an increase, becoming less negative. So that is what is going to happen over here, right? So injection of positive current will increase the membrane potential. So this will be the setup for that increase. One electrode will be used to supply the current, which is combined with the current generator. So a positive current will be sent inside, and there will be an amplifier which will be from with which we will be able to understand what is now the voltage inside, right? And if the current is provided, a positive current is provided, which is why the peak is in upward direction instead of the low, downward direction. Then, as a result of this, you can see there is an increased this thing voltage also, right? And sometimes the increase will be very minimal, but if the increase is such that it is able to cross a particular threshold, it will be able to generate something of a peak, which is kind of known as action potential over here, which we'll emphasize on later. The second question is, consider a cylindrical dendrite, right? You cut it along the length at two places to get a cylindrical piece whose total membrane resistance is R, right? So Rm over here is equals to R. Now you cut this along the length in the middle and get two cylindrical halves, right? What will be the membrane resistance of each half? Will it be R or R by 2 or R, 2R or R by 4? So since it is a numerical, I will let you look at it for a while, try to solve it, and then we'll come to the answer, right? So I'll give you some time. Just a moment. Yes. Yeah, I was facing some issue with my laptop. Okay, sorry, I revealed the answer, but I hope you too have been able to reach to that answer, right? So, okay, we'll discuss the solution for this one. The answer will be 2R, right? Why will it be 2R? Let's see. So, what does the first statement say? The first statement says that we have to consider a cylindrical dendrite, right? Something like this we will consider. Maybe a little shorter than this, which is easier to make, right? You cut it along the length at two places to get a cylindrical piece, right? So along the length, meaning like this, I will cut it at two places, right? Okay, along the length, meaning like this, I will cut it at two places, right? To get two cylindrical halves, right? So, wait, to get a cylindrical, cut at two places to get the cylindrical piece, whose membrane resistance is R. So, Rm now will be R. Now we have to cut this again along the length in the middle to get two cylindrical halves, right? So after the first thing, we got two halves like this, right? Yeah, somebody wants to speak, you can go ahead.
Ma'am, screen is not visible here.
The screen is not visible.
Yes, ma'am.
Is that the case for others also?
Yes, ma'am, it is not visible.
Okay, maybe I'll share again.
Ma'am, now it is visible.
Oh, it was visible. Okay, anyway, I'll share again. Okay, I hope it is visible right now. Right? So we are basically saying that after this first line, that is, we are cutting it along the length at two places to get a cylindrical piece, right? And the membrane resistance of this particular cylindrical piece is R, right? Then what are they saying? We will cut this again along its length in the middle to get two cylindrical halves, right? So something like this, right? What will be the membrane resistance of each half, right? So if this will keep on decreasing, so now this has been divided into two further parts, right? Yeah. And from the question itself, we know that this entire thing had a resistance of R, right? Now, when it will be divided into two parts, it will have a resistance double, like two equal parts we are cutting it in, right? So it will automatically become double of what it was earlier, right? Why? Because the resistance is going to increase if the membrane is going to be smaller, right? If Rm, this is Rm basically, right? So if the surface area is going to be smaller, the resistance is going to be more, right? So surface area will become half on cutting the cylindrical cylinder into two halves. Therefore, membrane resistance will become twice, right? Like I might not have drawn it that well, but if you recall Sir's lecture, that length constant, time constant lecture, in which this was there in the case of the length constant, wherein we were discussing lambda, how what is lambda dependent on? It is dependent on the membrane resistance and the axial resistance. So over here, this question discusses mainly about membrane resistance, right? So Rm over here is equals to R, right? And membrane resistance is inversely proportional to the surface area, right? So if there is more surface area, there will be less resistance. If there is less surface area, there will be more resistance, right? So that is why when we are cutting it into two equal halves, it is basically increasing, the resistance is becoming double of what it was before, right? The question could also have been framed in another way, that we have been given the, like after again cutting the previous one into these two halves, right? They are giving the resistance for one half, right? So they could also ask what was the actual membrane resistance of this bigger cylinder that was there, right? Now, what would that be? It would be R by 2. Okay. I hope that that concept I have been able to get across. There were so many glitches also during this. Yeah. Okay. Then this is again a theoretical one. A sodium potassium exchange pump brings sodium ions inside the neurons, brings potassium ions inside the neurons, brings both sodium and potassium ions inside the neurons, or releases energy in the form of ATP. So one of these is correct. The question is which one? Yes. So the correct answer for this one is basically it will bring the potassium ions inside the neurons, right? So this is an exchange pump. We know from what we have studied this week that there is more concentration of which ion within on the inner side of the neural membrane, intracellular side, which ion concentration is more? Is it sodium or is it potassium? Okay, so I'll say it is potassium that is more abundant on the intracellular side of the neuron, right? Extracellularly, which is more abundant, sodium. I hope I said potassium is more abundant in the intracellular side. Sodium is more abundant on the extracellular side, right? So when we talk about normal concentration gradient, it will favor the movement of potassium outside and sodium inside, right? Because the concentration of sodium is lesser inside, concentration of potassium is lesser outside, right? But over here, we are talking about pumps, not channels, right? Pump is going to use ATP, right? It is not going to release ATP, it is going to use ATP and it will use this energy to basically maintain this gradient as it is, right? So it will throw more sodium outside and more potassium inside. So that is why its function is to bring potassium ions inside the neurons, right? This is how it works, right? It looks more like the sodium potassium channel actually. But anyway, you will be able to still see, right? So this is the intracellular side of the membrane. This is the extracellular side of the membrane. You can see that there are 3 pockets for sodium and 2 pockets for potassium, right? So from the inside, it will take energy in the form of ATP and it will also take 3 sodium ions. What will happen because it has taken this ATP, it has now taken up this phosphate and only ATP left behind. This conversion caused, energy to be produced, and this energy it has used to basically change its conformation towards outside, right? Now outside, it will release whatever was there with it, and it will take more of potassium ions, and then again, it will flip because whatever cargo it had already it has been released. Now it will just flip and the new cargo will be released inside, right? And in such a way it will keep working. Yeah. So consider a neuron at rest. Let's say the equilibrium potential of the neuron for the ion potential for sodium ions for that neuron is E. We somehow magically swap the inside and outside concentration such that whatever concentration was present inside is now outside and vice versa. What will be the new equilibrium potential of sodium ion? Right? Will it be E or 1 by E or minus E or E by 2? So it is a simple question. Again, more based on logic than numericals, right? Which a lot of you were worrying about last week. So I hope this during, when I was saying the question out loud, you have already figured it out, right? I really hope that because it was, there are not much calculations that are required over here, right? So the answer actually is minus E. Why? What it is saying is a neuron is at rest. We know at rest the membrane potential of the neuron is generally a negative value, right? negative resting membrane potential or resting potential is there. Over here, we are talking about equilibrium potential though, right? So equilibrium potential will be different for different ions, right? So what is that potential for sodium ions? Yeah, I hope there was not a glitch. Just a moment, I'll change my network, I think. Yes. I hope it will work better over here. So what is being said over here is that we are not talking about the resting potential per se. What we are talking about is the equilibrium potential. Equilibrium potential will be different for different ions, right? Over here, which ion are we discussing? Sodium ion. Now, can anybody tell whether the actual equilibrium potential for sodium ion is positive or negative? Anyone? Okay. So in the during the end of the second week lecture, it has been discussed that the equilibrium potential for potassium is generally negative, right? It is negative 75 in case of squid giant taxon. For sodium, it is generally positive, right? It was around positive 55. Forgive me for the writing. That is the best I am able to manage with my mouse. Right. And chloride was also negative. Right. Over here, this is the equilibrium potential we are discussing. But like if we had to flip this, it will become negative minus 55. Right. But that is not what they are saying. They are giving assigning an equilibrium potential to sodium, right? So over here, we don't even need to remember this fact, right? So they are saying that we have assigned a equilibrium potential of E for sodium under normal conditions, right? Now, what if the inside of concentrations were swapped with the outside concentration, right? So everything inside the membrane, it was always negative. Outside the membrane, it was always positive, right? What if outside becomes negative, inside becomes positive? If that kind of a swap happens, such that whatever concentration was present inside is now outside and vice versa, right? What will be the new equilibrium potential of sodium ion? It will also be opposite, right? It will become minus E, right? So that is the answer. There is an explanation that is given over here. You can go through that as well. So if the concentrations are switched in the Nernst equation, ratio of concentration log inside the log, concentration inside the log becomes inverted or reciprocal, and therefore the final value will be negative of the original value. It will become like this, right? So after Nernst potential, this is kind of the value that we get, right? But it will become opposite to this. So this is for some particular kind of cell, not the squid giant taxon, the values, absolute values that were given. Question number 5 is, in a typical neuron at resting state, which of the following is most accurate? Potassium ions would like to move from the inside to outside according to the electrical gradient. Potassium ions would like to move from inside to outside according to the concentration gradient. Potassium ions would like to move from inside to outside according to both the concentration and electrical gradient, or potassium ions would like to move from inside to outside by none of the gradients. So which one is going to be true?
Ma'am, C.
Sorry, I did not get that.
Ma'am, see, potassium ion would like to move from inside to outside according to both concentration and electrical radius.
Okay. So in case of potassium, actually, that is not true. That will be true in some other ions case. In case of potassium, what is happening is uh, from inside to outside, it will move based on the, okay, negative it is. Yeah. Yes, that is true. So this will be the answer, right? Concentration gradient. Why? We'll see. So what is going to be the electrical gradient? It is going to basically determine how it is going to move in case of the lipid bilayer membrane, right? So this is a membrane, for instance, right? Membrane. The outside of the membrane is what charge? Is it positive or negative? It is going to be positive, okay? Outside the membrane. And inside the membrane, what is going to be the charge? It is going to be negative, right? And where are more potassium ions more abundant? Is it outside or is it inside? They are going to be more abundant inside, right? So this is out and this is in, right? So according to concentration gradient, what will be the direction of flow? It will be from inside to outside, right? Why? Because it is present in more abundance inside and in less abundance outside, right? But when we talk about the electrical gradient, right? Now, electrical gradient means what? They are what? Positively charged ions. Potassium ions are positively charged ions. So, will positively charged ion be moving towards positive potential or towards negative potential? It will move from positive to negative, right? To balance it out. So, over here, according to electric gradient, it will move from outside to inside. Even though their concentration is lesser outside, it will still move from outside to inside according to electrical gradient, right? So that is why only in case of concentration gradient, they will move from inside to outside, right? Yeah. So that is why B is the correct answer. So potassium ions are present in higher concentration inside, therefore they will like to go outside according to the concentration gradient. According to electrical gradient, however, they would like to move inside as the inside potential is negative. Okay. Now question number 6. Myelination of an axon increases its length constant. Is this statement true or false? So this is something that Sir had very subtly described as well in the same lecture in which length constant was actually introduced. Okay. So the answer to this one is true, right? What is myelination basically? It is kind of an insulating layer on the axon of a neuron, right? So axon is what? That long branch that is transmitting the signal from the cell body of a neuron to the dendrites of the other neuron, right? So it is basically that conducting branch. Now this conducting branch, if it has myelination, which is kind of like an insulating layer, if it has that, it is going to increase the length constant. What does this mean is, okay, first we can look at it theoretically, then I will explain it to you. So it increases membrane resistance and therefore increases the length constant, right? So if the membrane resistance is higher, less leakage of current will happen outside, right? If the membrane leakage is lesser, it will be leaky, right? So whatever current is touching the membrane, it can just leak outside. But if the resistance is higher, it will keep this inside the membrane, inside that cylinder itself, right? So a good example is a naked wire versus an insulated wire, right? If you look at a naked wire, if a current is flowing from point A to point B, and you basically just, cause anything to happen, you touch that wire, pour water over that wire, it will hinder the flow of current from point A to point B, right? But it will probably cause a short circuit, right? But if this wire is carrying current from point A to point B and it is insulated, right? If you are touching it, if small drops of water also fall on it, it is not going to affect the current that is flowing inside the wire. Why? Because it is well sealed and the resistance of the membrane is so high that it is not allowing the current to leak out or the outside thing to affect inside, right? So this kind of an insulation is going to help in, faster and longer conduction of signal, right? So because signal is not leaking, the length that it can transmit is higher now, right? So, yes, length constant depends on two parameters, Rm, which is membrane resistance per unit length, and Ra, that is axial resistance per unit length, right? So, this kind of an insulation will increase the length constant because it is directly proportional to the uh, length constant also, right? Which of the following is most reasonable value of time constant in neurons? Is it 10 minutes? Is it 10 seconds? Is it 10 milliseconds or is it 10 nanoseconds? Right? So we were seeing that in length constant, what could be the value? Like, if, for instance, we are talking about voltage, will it be in millivolts or nanovolts? If we are talking about amperes, will it be in, milliampere or picoampere? What will it be? And accordingly, this value will be determined. Right. So in case of time constant, there are two things that are affecting the time constant. One is the resistance, and the second is the capacitance. If you know the unit with which we in which we deal with this resistance and capacitance in terms of neurons, you will know the answer for this one also, right? And it has been discussed in parts, right? So the answer over here is 10 milliseconds, right? How? Because this tau, which is the time constant, is equal to RC, that is resistance into capacitance, and in a neuronal membrane, resistance is in a order of mega ohms, right? Because it is very tiny. These neurons are very, very tiny structures and they are very, very compact. So the resistance becomes higher, right? Because it is getting less surface area, less axial area, axial length, etc. So less diameter. So the resistance is in the order of mega ohms, that is 10 raised to power 6 ohms. While capacitance is in the order of nanofarads, that is 10 raised to power minus 9 farads, right? So when we multiply this R into C, we get 10 raised to power minus 3 seconds or 10 milliseconds, right? Consider a long cylindrical axon whose membrane potential is minus 60 millivolt at rest, right? Length constant is 2 millimeter. The constant hypopolarizing current is injected at point A in axon. When current is maintained for a while and a steady state is reached, which of the following point in axon will have least negative membrane potential, right? Will it be exactly at point A, or 2 millimeter left of A, or 4 millimeter right of A, or 5 millimeter right of A?
Ma'am,
Right?
question is not visible.
The question is not visible. Okay, I'll try again. I switch my net again, actually. I hope it is visible right now.
Yes, ma'am.
Okay. So we read it once more. It is a simple question, right? You just have to, again, it is a logic based question rather than a numerical based question, right? You don't have to do a lot of calculations over here. You just have to think what is logically going to be the answer. Consider a long cylindrical axon whose membrane potential is minus 60 millivolt at rest. So they have given you a resting membrane potential, right? They have given you a length constant which is 2 millimeter, right? A constant hyperpolarizing current is injected at point A in the axon. There is one point wherein a constant hyperpolarizing current is being injected, right? When the current is maintained for a while and a steady state is reached, which of the following points will have a least negative membrane potential, right? So the only point that you need to know over here is what is a hyperpolarizing current and what is the length constant. If you know these two things, you will be able to answer this. Okay, so let us discuss. Before we come to the answer, let us discuss it, right? Let us arrive to that. So they are saying that the resting membrane potential is what? It is minus 60, not Rm, sorry. It will be very confusing if I write this also as Rm. Let me write it as Rp. Rp. Hold on. Lots of mistakes I have made. Yeah. So they are saying that this minus 60 millivolt is the resting membrane potential. Okay. And they are saying that the length constant, that is lambda, is this 2 mm, right? And a constant hyperpolarizing current is injected. Now, what is hyperpolarizing? What is the polarized state in this case? If the resting this is 0, right? This is positive value, positive say 50, and this is say negative 60, right? They are saying that the resting membrane potential is this negative 60, right? So the normal polarized state is what? It is minus 60, right? Now, what will be hyperpolarization? It will be going further down, right? Further down. What will be depolarization? It will be coming upward, right? Towards 0. If we come towards 0, it will be depolarization. If we go further away from 0, it will be hyperpolarization, right? So over here, they are saying that a constant hyperpolarizing current is injected at point A in the axon, right? So there is a point in axon with a normal resting membrane potential of minus 60, wherein a hyperpolarizing current is injected, right? So wherever that point A is there, if a hyperpolarizing current is injected, what will it become like? Its potential will become like this, right? It will become hyperpolarized. It might go to minus 70, say, for instance, right? It can come till here, right? So it is hyperpolarized now. Now, what are they asking? That this effect of hyperpolarization, how, like, what is the point over here which will have the least effect of hyperpolarization, because they are saying least negative membrane potential. We are going towards more negative membrane potential, right? So the point right next to this A will also somewhat go to a negative potential, right? Adjacent to that, it will also somewhat go to a negative potential, maybe just 61, 62. After that, it might not change at all. It might be still minus 60 only, right? So the farther away from point A it will be, the less negative membrane potential it will go to, right? Because the less effect of this hyperpolarizing current it will have. Okay. So what will be the answer over here? It will be 5 millimeter right of A. Whether it was even left of A, it would still have been this answer because it is farther away from point A, right? Now, this question also could have been stated in a different way, right? They could have written that a constant depolarizing current instead of hyperpolarization, they would have written a constant depolarization current is injected at point A in the axon, right? When is the current is maintained for a while and steady state is reached, following which of the following points will have least negative membrane potential? Now, what will be the answer? If you have got the crux of this, you will be able to answer this, right? Again, I'm drawing the same graph. I would really love it if one of you would arrive at the answer. This is the resting membrane potential again, right? And say where point A is, it is over here, right? And what they are saying that instead of hyperpolarization, they would have said that depolarizing current is injected, right? Now, what will this depolarizing current look like? Depolarizing current meaning a positive current, right? Which will make its value go near to zero, right? Now, where will this value go near zero maximum? That is least negative membrane potential. Which one it will have? It will be exactly at point A, right? Then its neighbors will have a little more positive value than the resting membrane potential. Its neighbor will have a little more positive value than resting membrane potential, but the least value or the maximum away from resting membrane potential and towards zero will be exactly at point A in case the question was framed in this way, right? Yeah. Next question. So as we'll just read this concise explanation also. As the current is injected in the hyperpolarizing, that is, current injected is hyperpolarizing, which means negative. Membrane potential at point of injection A will be most negative, right? The effect will reduce as we go away from A. So the farthest point will have least negative membrane potential. So farthest point over here is 5 mm, right? Question number 9. Consider a neuron in the default resting state. Now, a constant current stimulation is applied in the neuron. In this initial period of stimulation, the current will mostly charge the membrane. The current will mostly flow through the ion channels. The current will mostly charge the ion channels, or the current will mostly flow through the ion exchange paths. What will happen?
Ma'am, the current will mostly flow through the ion exchange form.
Okay. But this is during the initial period of stimulation. I would like to, that is the key point. So exchange pumps will actually come the last in picture. Okay, when everything depolarization, repolarization, hyperpolarization, everything has happened. Now to maintain an equilibrium, ion exchange pumps will come. But during the initial period of stimulation, what will happen? It will mostly charge the membrane, right? First, that capacitance will build, then transfer will start happening, right? So just before you were taught that this time constant and length constant, there was that I don't remember the title of that particular presentation that in sir's lectures, right? So what will happen? It was given that suppose this is a resistor, this is the inside, this is the capacitor. That straight line has gone by mistake only, okay? And this is the outside, right? So when a current is actually passed, first will it pass through the resistor or the capacitor? First, so it will depend on the voltage that is there, right? So currently, initially, during the initial period of stimulation, both will not have a potential difference, right? So potential will be zero over here also, here also. It will start passing through capacitor, right? Once over here, this charge is built, and consequently over here, the okay, really facing trouble with the internet today. Yes. So due to this, what will happen? First, it will charge the membrane through this, right? And then other things will start happening. And the ion exchange pumps will come last of all, right? When the membrane potential has to be equal, taken back to the equilibrium state. So then this particular state will come. Sorry, this one. Yeah. Okay. If I would have remembered that I had this over here, I would not have drawn it over there. So the capacitive part of the membrane offers no resistance, right? So over here, there is no resistance whatsoever. The current will initially go through the capacitor to charge the membrane, right? So the natural flow of current will be first over here. Once a voltage gets built up, then it will come over here also. And then through both sides, it will basically flow towards the outside, right? Now question number 10, the final one. The resting membrane potential does not depend on A, temperature, B, concentrations of ions with non-zero permeability, C, concentrations of ions with zero permeability, or D, whether the ion has positive or negative charge. Right? So which one of the following does the resting membrane potential does not depend on? So if you remember the equations, the Nernst equation, the other one, this
Temperature.
temperature, okay? But the Nernst equation does have that RT in it, right? Temperature was there, a part of it. Or was it in the equilibrium potential? We'll see. So it is the concentration of ions with zero permeability actually, right? So the resting membrane potential is not dependent on that. So ions that are not permeable do not affect the membrane potential at all, right? Because they are not going to go inside or outside. And it can also be seen in the GHK equation, right? So GHK equation does have that T, the absolute temperature, wherein we will change the temperature to Kelvin, right? And the gas constant, right? So that RT that is there in the GHK equation. So temperature is going to affect, concentration of ions will affect, and the charges of ions will also affect. But if the ion is not permeable at all, it will not affect, it will not have anything to do with membrane potential, right? So ion concentrations are multiplied by permeability, and therefore ions with zero permeability will not have any effect over here, right? Okay, so I will just switch my presentation. Okay. It's really hard to keep track today whether I am online or not. Yeah, so I'll share the other presentation that we had left off in the previous, midway in the previous one, right? We switch to that. Yeah, this. So a great deal of this we had already gone through. fMRI also we had seen, right? Now from here onward we get to discuss, right? The different techniques that are used to study the brain is what we were discussing. Okay. So over and I'm discussing similar techniques together, right? So for instance, CT scan, MRI, fMRI, I was discussing together. Now MEG, ERP, EEG, I'm discussing together, right? So the first one over here is MEG, right? And these are all interrelated, right? That is why I have pulled them and kept them together. Their principles are very, very similar, right? So MEG or magnetoencephalography and event-related potential, that is ERP, right? So MEG is a non-invasive medical test that measures the magnetic field produced by your brain's electric current, right? So what is going to happen? We have seen that there are electric currents that are going on, right? Be it synaptic transmission, which we'll study later, be it this action potential generation, right? The resting membrane potential of the brain, the hyperpolarization, depolarization, etc. So there are a lot of electric currents that are going on inside the brain. And you know there is a close relationship between the electric field and the magnetic field, right? So if one is there, the other can be also determined, produced and determined, right? So over here, from the brain's electrical currents, magnetic fields are determined and they are basically measured, right? It is performed to map brain function and to identify the exact location of source of epileptic seizures generally, right? So last time we have already discussed what is epilepsy, what are seizures, right? So MEG is also more commonly used in those kinds of diseases and identification of the exact, focal point of the origin of these kinds of seizures. Then event-related potential. The name itself suggests that we are calculating a potential that is related to some event, right? It captures electrical activity in the brain at specific points relative to an event or a stimulus, right? So this event could be, something from within, such as an epileptic episode, a seizure, right? Or it could be like we are giving, because we are measuring this ERP, we are first giving some stimulus from outside. We are showing certain kinds of pictures to you. We are, tickling the subject, or we are causing, we are giving them very mild electric shocks, and what is the response and where that response is coming is basically being studied by this event related potential, right? It is measuring the electrical activity itself. Then there is electroencephalogram, right? This is the EEG test. Now I'm sure most of you might have heard about the EEG. If not had somebody around them go through an EEG test, you might at least have heard about this, right? It is almost as common as an ECG or electrocardiogram, right? So this is a test that measures electrical activity of the brain, right? What it will do is it uses small metal discs called electrodes, which are attached to the scalp, right? Now, this is actually the older method. Now they have a cap sort of structure, and that cap is put on your scalp, right? Instead of directly attaching electrodes to your scalp, they are putting that cap on your scalp, and in between the electrodes and your brain, they like that cap has a hollow region inside, wherein a kind of gel is put, right? For instance, when we go for an ultrasound, they put the gel and then they, look around. So it facilitates the ultrasound waves to basically pass through. Similarly, a gel kind of is put, and it is not directly on the scalp over here, right? Brain cells communicate via electrical impulses. We know that neurons communicate via action potentials, right? And this activity shows up as wavy lights on EEG recordings. So brain cells are active all the time, even during the sleep, right? The pattern of these waves in EEG might vary depending on whether a patient is awake or the patient is, doing some very, very attention, demanding job, or they are sleeping or they are dreaming. It might vary. So this is the principle of MEG, wherein electric current that is, being generated by these action potentials and all, it is being converted to magnetic field, which is being read, right? So these are the sources of magnetic field, right? The electricity that is going to get transferred from different parts of the neuron to other neurons, and orientation of neurons. You don't really need to go into this much of a detail, right? But this is what the output actually looks like. Then this is ERP, right? Wherein again, electrodes are recording something, but it is related to some stimulus that we are providing, right? And this is the kind of output we are getting, right? And this is EEG. Like I mentioned, there is a cap these days that is present, and there are electrodes on the cap instead of directly on the skull, right? And these kinds of waves are actually produced. Okay. Now, these kind of waves that are produced, they are then refined to understand what is, what is the kind of, uh, status, brain activity status of the subject that we are studying. Yeah. Then we come to optical dyes. Right now, this again is a different kind of a technique. Over here, what happens is optical imaging via multi-photon microscopy can measure activity-dependent changes in fluorescent signals directly from individual neurons, astrocytes, and blood vessels in the living brain itself. Right? There are some variations to it, one of which is voltage sensitive dye imaging, which is VSDI. It is an accurate imaging method that uses small intensity changes of fluorescent dye emission to capture the brain activity, right? So what will happen is we are injecting basically some fluorescent dye, right? And wherever the brain activity is happening, right, it is going to utilize something and it is going to release some CO2 and stuff, right? This chemical change will be basically visible by the color change in this fluorescent dye, right? Which we will be able to capture via imaging. So that is how this particular technique works. So this is what it is more commonly used in lab subjects than actually in the pathological or diagnostic conditions. This is how it happens, right? So voltage sensitive dyes are there, which will emit signals, right? And you will again record it, and you will know that, okay, this is the region of the brain that is more stimulated or it is functioning, right? And this is the region of the brain where the stimulation is not happening, right? Again, these are the kinds of microscopes but you need not go into this detail right now. If and when we come to, understanding the different kinds of sections, how, what are the kinds of staining materials of these ex vivo sections of these brains or autopsied brains or the animal brains, right? Then we can discuss this portion also, right? For now, I'm going to skip that. Then another important technique, and which is very, very temporally efficient, but very, very invasive, right? It is both temporally efficient and, spatially also efficient, because it is going to tell us the recording of a single neuron also, right? And in the order of milliseconds itself. But it is very, very highly invasive. That is why it is not that commonly used. It is also mostly used in research settings, in laboratories only, right? So it is a single unit recording, and the patch clamp in single unit recording is a precise method of recording the electrical activity of individual neurons in the brain. It involves using fine wire electrodes to capture axonal spikes from neurons in response to various stimuli such as visual, auditory, or somatosensory inputs. Right? So this is how it happens. This is the EEG, right? EEG is from where? It is from the top of the skull, right? So this is the neuron. Then there is the skull also. On top of that also there is the skin. And on top of that, the electrode for EEG is there. Then there is ECOG. What is ECOG? That you drill a hole, not through the skull, but you drill a hole till the skull. You form an opening till the skull, and then you basically place this electrode and you take the recording from the surface of the skull, right? Or just below the skull, right? Then there is this FPN unit spikes. What happens over here is that you put the electrode deeper into the brain such that one particular bunch of neurons that is present in that particular area, if they have any changes as a unit, right, as a team over there, that cumulative changes can be captured via this unit spikes. And then there is intracellular recording, which is something that is single unit or patch clamp, in which a single neuron inside that you are introducing or in the electrode, and you are recording from within that particular neuron, right? The patch clamp is a technique is the generally recognized approach to study the ion channel function. During a patch clamp recording, a micropipette container containing a recording electrode connected to an amplifier and filled with electrolyte solution is brought into contact with the membrane of the isolated cell, right? So this is what it looks like, right? Cell attached recording method. So this is a micropipette which contains an electrode, and it has within it the electrolyte solution, so that the osmolarity will be maintained, right? If the channel is open, this kind of a recording is produced. If the channel is closed, this kind of a recording is seen, right? So based on this, we have already told that this final two techniques that we study, the patch clamp and the single unit recording, they are both temporally and spatially the most efficient ones. But why are we not using it? Because it is very, very invasive, right? So which will be an ideal technique to be used? Something that will have a millisecond temporal resolution, something that will have a micron spatial resolution, that it will be able to zoom into the micron level of everything. It will have unlimited bandwidth, right? And the data capturing and all will not be a problem, and no, it will be non-invasive, right? We can skip this part. You have already gone through this. This is basically the overview of what you had been taught in the first unit, right? So, because we had not finished this slide. This I would like to emphasize on, that there are two types of cells in the brain, right? Two broader categories of cells in the brain. One category is the neurons, which we talk of as the functioning units of the nervous system, and the glial cells, right? But I would like to tell you that glial cells are just as important, right? In fact, new functions are still being developed, discovered for these glial cells, various glial cells, right? So these are the major kinds of glial cells that we have inside the central nervous system, that is the brain and the spinal cord. We have these cells, which are the ependymal cells, the oligodendrocytes, which are going to produce the myelin sheath, basically astrocytes, which have multiple functions, and microglia, which are majorly the immunologically, act as macrophages. Then in the peripheral nervous system, we have the satellite cells and the Schwann cells, right? Schwann cells are going to act similar to that of oligodendrocytes in the central nervous system. So first we begin with microglia. Microglia are brain's immune cells. They serve to protect against injury and disease, right? Then we come to astrocytes. They are star-shaped cells that maintain a neuron's working environment, right? They are very essential for the workings of a neuron. They do this by controlling levels of neurotransmitter around the synapses, controlling concentrations of important ions like potassium, and providing metabolic support, right? Along with that, there are other things that they do. These are the ones that are responsible for, also they are a part of blood-brain barrier, right? If these are not there, then the blood-brain barrier is not completely made, right? It will still be leaky like in all other organs. So the brain will not have a specialized blood supply, which is an additional barrier, which is known as the blood-brain barrier, right? They are very crucial components of that as well, right? And they also help in, inflammation, right? If inflammation goes up in the brain, they divide to counter that, right? Then the macroglia. One of the macroglia is oligodendrocytes. So oligodendrocytes provide support to axons of neurons in the central nervous system, particularly those that travel long distances within the brain. They produce a fatty substance which is known as myelin, which is wrapped around the axons as a layer of insulation, right? Then are the ependymal cells. These basically line the spinal cord and the ventricles of the brain and are involved in creating cerebrospinal fluid or CSF, right? So this is what will secrete the CSF, ependymal cells. Then there are radial cells also that are present. These radial cells, these are the radial cells basically. They are the progenitor cells that can generate either the neurons or the oligodendrocytes or the astrocytes, right? Because they are progenitor cells. Then when we talk about the PNS, as I said, Schwann cells are similar to oligodendrocytes in the central nervous system. It myelinates neurons in the peripheral nervous system. Then there is satellite cells that surround neurons in the sensory, sympathetic and parasympathetic ganglia and help to regulate the chemical environment and may contribute to the chronic pain condition over there, right? Then we also have some other glial cells. For example, one is the enteric glial cell. As the name suggests, it has something to do with the enteric system, that is the digestive system. So these are found in the nerves in the digestive system, right? Then there are different kinds of neurons, right? The neurons can be unipolar, multipolar, bipolar, or they could be of pyramidal or Purkinje type, right? So dependent on different kinds of classification, neurons can be of different types, right? This is one representation of why different kinds of neurons are required, right? And yet another classification of neurons also, wherein they are classified as sensory, interneuron and motor neuron, right? And it also describes very beautifully why different kinds of neurons are required, right? So there is one neuron that is taking information from, the site of injury to the brain. There is one that is relaying the information from this sensory to the one that actually has to take decisions and to act. And then there is a third one that has taken the decision and is transmitting this decision to that particular site of injury, right? So that this process is quickened. If one person was running, for instance, if you, you know, look at this 800-meter race, right? Or you look at a marathon versus a 1 kilometer marathon versus a 1 kilometer relay race, right? Which one do you think will end fast? So there are more chances that the 1 kilometer relay race will end fast, right? After every 250 meters, there will be a new person who will do the job, right? So the person will not get tired. The person will have the efficiency to work better and will be able to, transfer this information, receive whatever was coming and transmit it faster, right? Because it is not fatigued. Okay. So, and the resources that we will spend will also be lesser in that case, right? We will not be providing water after 250 meter to this person. We'll quickly have to drink water and again. Nothing like that is going to happen, right? So, a relay race will be more efficient, time efficient, right? So, that is the same concept over here. Then there are synapses, right? We will discuss later more about synapses during the further units of this particular course, that is neurobiology. So, for now, what you need to know is what exactly a synapse is. That is all we are going to talk about right now. So, it is basically the junction between two neurons, right? So these two neurons are basically the neurons that will connect or will transmit information from one to the other, right? And usually it will be from the axon of this first neuron to the dendrite of this second neuron. So this junction that is between this axonal end feet and this dendritic branches, this particular junction is known as synapse, right? This is the place wherein neurotransmitters are released from the axonal ends and taken up through the dendritic ends, right? And this is a nerve circuit. We'll discuss it later. So then there are some common terms that I would like to discuss, right? These common terms are something that people can tend to get confused with, right? One of them is the white matter and gray matter, right? What is gray matter? It is basically made up of cell bodies. And what is white matter? It is made up of myelinated axons, right? So, how can you simply remember this? You can remember that because we have studied when we were looking at oligodendrocytes that this myelin is basically a fatty layer, right? Fat generally in the body is white in color. Okay. Now I find this way easy to study because I do a lot of dissections. So I look at a lot of fat and I know that it is white in color, right? So it is easy for me to remember that, okay, that white colored thing will have fat in it, so it will have myelin, right? So the myelinated part of the neuron will be the white matter, and the cell body of the neuron, which is not myelinated, will be the gray matter, right? So that is how I relate to it. But if you have another way of remembering it, uh, then that is also fine, right? Then there is nerve and tract, right, which is essentially the same thing. Just the difference is, okay, I have made a mistake over here. I'll just correct it over here itself, right? Oh, sorry. Yeah. So this is essentially the same thing, but you generally would have heard about nerve and not tract, right? So what is a nerve? It is a bundle of axons found in the body. And tract is the same thing. It is a bundle of axons, but when these bundle of axons is found in the brain, in the CNS itself, it is known as a tract. Similar for a ganglia and nucleus, right? So when a group of cell bodies is found in the body, it is known as ganglion. But when it is found in case of the brain, it is known as a nucleus, right? Then there are different kinds of nervous system. The central nervous system, which has a brain and the spinal cord. The peripheral nervous system, which is basically everything outside of the brain and the spinal cord, this peripheral nervous system is further divided into the afferent and the efferent kind of nervous system. Afferent has more to do with sensory division, right? So it will sense external environment, it will sense internal environment, and it will basically take this information either only to the PNS or sometimes also take help from the CNS also, right? And the other is the efferent division, which will basically further be divided into autonomic nervous system and somatic nervous system, right? This autonomic nervous system, for instance, is the heart, right? So it has something to do with the motor function, right? The beating of the heart, the heartbeat, all of this is dependent on the autonomic nervous system of the heart itself, right? Similarly, there is somatic nervous system also. Then there are certain layers of that protect the brain, right? So this is the brain. There are three meninges that protect the brain, right? These are the pia mater, arachnoid, and dura mater. On top of that is the skull, which is a bony structure, and then the skin, right? The hypodermis and the dermis. Then we can talk about CSF, right? Again, a lot of people I have seen that even after discussion about, because during, normal neurobiology lectures, we tend to skip. We know that there is CSF, right? We know that there is blood-brain barrier, but we don't know what is blood-brain barrier. We don't know what is CSF, right? What exactly is it? If we talk about the composition, for instance, what is it? So that is why I have kept the slide. Right, what is cerebrospinal fluid? It is an ultrafiltrate of plasma which is contained within the ventricles of the brain. What were the ventricles of the brain? Those hollow regions of the brain. Right, they were hollow because they had this fluid in them. And the subarachnoid spaces of the cranium and the spine. It performs vital functions including providing nourishment, waste removal, protection to the brain, etc. The adult CSF volume is estimated to be around 150 ml with a distribution of 125 ml with arachnoid spaces and 25 ml within ventricles, right? This CSF is predominantly secreted by choroid plexus with other sources playing a more poorly defined role, right? So it's not yet studied that well. That is why it is written more poorly defined. It might be there, but we don't know it as yet that way, right? So this was just completion of last time's lecture, right? Okay. Yeah. So we can just begin at least with the second week's discussion also. We'll see how much we want to go into detail for this one, right? Actually, this still has some part from week one that is still there, right? Because in week one, I felt a lot of introduction was there. So a lot of things tend to be, introduced on the surface. So I wanted to, uh, spread a little more light onto that. So I'll just discuss at least the part that is pertaining to lecture unit one, right? For instance, the brain orientations I wanted to go through again, right? So, because this is something that I feel once or twice you have revised it, once or twice you have understood what it is, you will not forget it for life, right? And this is not just for brain, it is for other organs also, right? What is posterior, what is dorsal, what is ventral, you will remember, right? So, the first part that I always say is you should know what is dorsal ventral, right? And that not just in case of the brain, you should know it in case of your body. You should, even if some other organism is being talked about, you should know what is the dorsal side, what is the ventral side, right? Because it is that simple to remember. So, what is dorsal? It is basically sun facing, and what is ventral? It is basically away from the sun, right? Now, why am I saying sun facing and away from the sun? Because I am standing straight. Sun could be over here also. It could be over here also, right? So this I am saying with respect to us not being bipedal, right? Meaning, what if we were not standing on 2 feet, right? How would we crawl? right? For instance, babies, how do they crawl? They crawl on their four limbs, right? So when you come into that position, or most other organisms are already in that position, right? The dog is already walking on four limbs, right? The cockroach is walking on its limbs, right? Uh, so in all organisms, including us, the dorsal side is the back side, which is basically going to face the sun, right? The top side or the back side, whatever you would like to call it. The side which is going to face the sun is the dorsal side, right? So, for instance, the dorsal side of my lung will be this, my lung from this side, right? And ventral side of my lung will be my lung from the front side, right? So, similarly, like, if, for instance, we were just going through a, physiology class, I would say that stomach is towards the ventral side because it is towards the front, and kidney is towards the back, right? So it is towards the dorsal side, right? So this is same for all organisms, right? In the cockroach, the exoskeleton is towards the dorsal side. The wings are towards the dorsal side. The ventral side has the feet, right? So it is easier to remember. That is why I'm beginning with this, right? So dorsal, ventral. In case of brain, what will be dorsal? This part will be dorsal. This part will be ventral. Because if I were crawling, this will be facing the ground. This will be facing the sun, right? So this is dorsal. This is ventral, right? Now, what will be anterior, posterior? Anterior is always the front, posterior is always the back, right? This is like English itself, right? In English also, that is what it means. So, anterior side is also often known as rostral side, right? Over here, it is easy to remember in case of brain because what does rostral mean? It means nose, right? So, the side that is facing the nose is the front side. What does caudal mean? Caudal means tail, right? So the side that is facing the tail towards the back is towards the back. It is like directly opposite of rostral. Is over here. It is the caudal side, right? So dorsal, ventral, rostral, caudal, right? And then there is one other thing, and it will depend on organs, right? For instance, in case of this dorsal, in case of spinal cord, if we are looking, then the rostral will again be towards the nose. This part of my spinal cord will be rostral, and towards my tailbone, it will be caudal, right? Because caudal actually means tail, right? Then there will be this other thing, which again is simple English, medial and lateral. Lateral on the side, medial in the middle, right? So that is how we remember these brain orientations. So I'll change the slide and I will just come, I'll just plug in my laptop. One moment. Yes, I share again. Okay. So this is what we were discussing, right? We'll just read it quickly. Anterior is in the front or towards the face. Posterior is behind or towards the back. Superior is above towards the head. Inferior is below and towards the feet, right? Medial is toward the middle. Lateral is towards the edge, right? Towards the side. Dorsal is towards the top of the brain or the back of the spinal cord. And ventral is towards the bottom of the brain or the front of the spinal cord, right? Rostral is towards the front of the brain or top of the spinal cord, and caudal is towards the back of the brain or the bottom of the spinal cord. So if you read it like this, it might sound a little difficult, but I hope when we were going through it, just with the diagrams, you have understood, right? Then these are the various ways in which brain orientations are basically studied, especially when we are looking at autopsied brain samples, or we are sectioning, the brains in the mice brain or the rat brain in case of a laboratory setting, right? So these are the various orientations. One is the frontal or coronal plane, right? What does it mean? That if this is the brain, it is being cut like this, right? That is coronal. What is the sagittal one? That if this is the brain, then it is being cut from the middle, right? One half and the other half. And both the halves will be identical to one another over here, right? Like it will have all the brain regions and the other half will also have all the brain regions, right? In case of the frontal or coronal, that is not usually the case, right? Like if this is the region, then only the temporal and this temporal and frontal lobe, these regions are coming, right? No parietal lobe, no occipital lobe is coming, right? Over here, like in this one, all lobes are more or less covered, right? Unless and until they are on the lateral side and you are doing it from the middle, right? But more, it depends on the kind of thing you want to look at, actually, that which kind of section you will go with, right? Then there is horizontal plane, which is basically instead of like this or like this, you are cutting the brain like this. That is horizontal, right? Then when we talk about structures of the brain, there are these three major parts of the brain, right? They are the forebrain, which is the largest part of the brain, the midbrain, and the hindbrain, right? When we talk about the forebrain, it includes structures like cerebrum, thalamus, hypothalamus, pituitary, limbic system, and olfactory bulb, right? The midbrain consists of various cranial nerve nuclei, that is a bunch of cell bodies of the cranial nerves, the tectum, tegmentum, colliculi, and crura cerebri, right? And the hindbrain, which is also referred to as the brain stem, this one, is made up of medulla, this thing, pons, cranial nerves, and back part of the brain, which is known as the cerebellum, right? So this is these structures in detail, right? This is cerebellum, this is spinal cord, medulla, pons, midbrain, pituitary, then hypothalamus is right there above it, the CG, corpus callosum, thalamus, pineal, these you don't need to remember right now, right? These are the major parts of the brain which are made up of different structures and have important and different functions also, right? So first we will talk about the forebrain, right? So we'll go through one by one through these different various major parts of the forebrain. One of them is thalamus, right? Now, what does the thalamus do? It is present near the midline of the brain and like it is present towards the middle of the brain. If I cut sagittally, it will be there, right? They are known as a relay center of the brain because of all the sensory input that is coming into brain from various sensory organs that passes through thalamus except olfactory senses. All motor information goes from the brain to thalamus and then to the rest of the body, right? So it is relaying information. It is bringing sensory information from the body to the brain and motor information from the brain to the body, right? So this is a place wherein that exchange is happening. Then we come to hypothalamus. Now, hypothalamus is located above the pituitary gland and below the thalamus. It controls the pituitary gland, which controls all the endocrine glands in the body. And it is responsible for body temperature as well as motivational behaviors like hunger and thirst, right? Then comes hippocampus. It is a part of the limbic system. It is located in each temporal lobe and is critical for learning, memory formation, as well as spatial navigation, right? It is actually a very crucial, very small, but very crucial part of the brain. Amygdala is also a part of the limbic system. It is just present in front of the hippocampus only. It is at the end of the hippocampus. It is responsible for the response and memory of emotion, especially the emotion of fear, right? Then comes the basal ganglia, which is important for regulation of various motor functions, and cerebral cortex. The outermost and topmost layer, this thing of the brain, it is divided into frontal, parietal, and temporal and occipital lobes. We have already seen that, right? The cerebral cortex is responsible for thought processes such as speech decision making. Each different lobe is responsible for different cognitive and processing functions, right? And this part actually we have already studied in Sir's lectures. That what are the functions of the parietal lobe, occipital lobe, where is the sensory processing area, where is the sensory cortex area, motor cortex area, etc. Right? And together, basal ganglia and cerebral cortex form the cerebra. Right? Then comes the corpus callosum. So you might remember this. It is basically the junction that is basically bridging the two halves of the brain, right? So it is located above thalamus, under the cortex, connects the right and left hemispheres of the brain, and is responsible for communication between the two hemispheres of the brain. It is also very crucial. If this was not there, uh, then actually part of your body will not know, and part of your brain will not know what the other part of the body and the brain are doing, right? It is that crucial. And there have been instances where people have damaged or missing corpus callosum, and this kind of a thing has happened with them, right? Then comes the midbrain. It is involved in vision, particularly visual reflexes, hearing, sleep-wake cycle, alertness, and temperature regulation. Then comes the pineal gland. So this is located in the center of the brain and in the midline between the two hemispheres. Part of the endocrine system produces melatonin, which helps maintain circadian rhythm and regulate reproductive hormones. It also regulates the endocrine functions and converts signals from nervous system into endocrine signals. So it is basically the hub wherein this endocrine signaling is happening, and or it is somewhat responsible for some part of that endocrine signaling. Then we come to the hindbrain. So the major part of the hindbrain is the cerebellum. It is located in the lower area of the brain behind the pons. It is responsible for balance and coordination of muscles of the body, specifically the involuntary responses. Then comes medulla oblongata. It is located in lower part of the brainstem. It is responsible for maintaining all vital functions of body and involuntary actions that sustain life, like breathing, blood pressure, heart rate, swallowing, and transferring messages from the brain to the spinal cord. Then comes pons. It is located above medulla oblongata. It is responsible for sending signals or messages between several areas of the brain, especially between the upper and lower parts of the brain, and plays a role in sleeping as well as in dreaming. Right? So these are the major lobes of the cerebral cortex: frontal, parietal, occipital, and temporal lobe. When we look from the top, then we cannot really visualize the temporal lobe because that is towards the lateral and ventrolateral side, right? And I have already told you what is sulci, what is gyri in the previous section. Right? These are the various lobes of the cerebral cortex and the sub, division of the regions of these lobes, which also you have already looked at. Then I hope you are aware about the somatosensory cortex and the motor cortex, right? So if you go back, in this parietal lobe and frontal lobe, there are regions that are this, region for motor processing. And before that, the region which is relaying this information or bringing about this information, which is the motor cortex region, and somatosensory cortex, which is getting that information into the brain, right? The information that this motor cortex is responded to, right? So these are basically these strips, right? The pink one is the motor cortex, the blue one is the somatosensory cortex, and these are basically the homunculi, regions that are allocated for different kinds of functions or different kinds of organs, right? So over here, for instance, you can see a great deal of region is given to abdomen, pharynx, right? And tongue, jaw, teeth, gum, lips, etc. Whereas this is in case of somatosensory, whereas the arm does not have that bigger region, right? Because not many senses you have to, feel on the arm, but much more senses you have to feel in your fingers, your hand, your tongue when you are tasting things, uh, etc. Right? The leg, the hip, the trunk region, you are not feeling that much, right? Similarly, you are not performing a lot of motor functions with this, right? Arm, you are just moving your joint, right? Arm, particularly, you are not moving. So that much region of this motor cortex is not dedicated to the arm or the hip or the trunk or the shoulder, right? Just the joints, right? And these other areas which have something to do with fine functions, or the tongue constantly, even if you are not eating anything, it is continuously moving, because you need the tongue to move even for you to speak, right? Similarly, your lips, your eyes are always moving, right, when you are awake, etc. So these have been allocated more area in this motor cortex region than these other regions that do not have those many motor functions linked to them, right? And this has been inspired by the work of Wilder Penfield, about whom I have explained in the previous class, and the models that are based on the sensory and motor cortex area and the region of organs that have been allocated to these cortices are basically looking like this, and they are known as the sensory homunculus and the motor homunculus, right? Then these are what the human brainwaves look like, right? These are the major categories that these brainwaves lie in. There are five major categories. The fast brainwaves are the gamma waves. The slower than that are the beta waves. Slower than that are the alpha waves. Then after that comes the theta waves, and the delta waves are the slowest waves, right? Over here, delta waves are generally achieved during dreamless sleeps or loss of bodily awareness or repair function when the body is undergoing repair, right? Theta waves is generally, achieved during insight, creativity, dream, deep meditative states, reduced consciousness, in those states, right? Over here, the brain is relaxed in the delta and theta phase. Then comes the alpha phase. This is when you are physically and mentally relaxed. You are awake, but you are relaxed, right? Beta is when you are awake and you are alert and conscious, and you are constantly thinking or you are excited about something. Gamma waves is during heightened perception, learning, when you are solving some complex problems, when a lot of cognitive processing is being done in your brain, then your brain is active, and it has these high frequency waves, which are the gamma waves, right? Uh, Prolonged heightened waves, or waves that are of even higher frequency, is that uncontrolled high brain waves, which could cause epilepsy. So learning, problem solving will not cause that. That only occurs when there is an innate flaw in the brain itself, right? In some functioning is going haywire in the brain, then only that will happen. So, information is transferred within a neuron and between two neurons by either electrical or chemical signals. We have already studied this, right? This is an example of electrical activity, right? There is a sequence of spikes that you can see over here, which are also known as action potential. So, this single one is known as single action potential. So what gives rise to this electrical activity in the brain, right? This you have already gone through. It is the movement of charged particles that is happening over here. Now, what are these charged particles? Are they electrons? No. Over here, they are ions, right? These are ions such as sodium, potassium, chloride, calcium, magnesium, etc., right? So what makes these ions move in the brain? right? So these ions move due to various variations of concentration gradients and electric field, due to which they are, there is a favored direction of their movement. Either they will move from in to out or out to in, and these are the mammalian cell concentrations of each of these ions, right? The major monovalent ions include sodium chloride and potassium, and the divalent cations, which are the minor, like they are present in much less concentration, they are the calcium and magnesium. So these are the major ones that are there in the body and the mammalian cells. And this portion also you have gone through, that what is actually preventing these ions from moving out through the membrane and balancing out. It is the lipid bilayer membrane, which is hydrophobic, which prevents the ions from co-crossing, right? Only hydrophobic and small hydrophobic molecules can cross through this membrane efficiently. Otherwise, there is a barrier that it creates. So how do these ions cross these membranes then, right? It is by the presence of ion channels. These are specialized passages that are selected for particular ions and let them pass through the plasma membrane. These are the various kinds of ion channel receptors in the body, right? They could either be voltage gated ion channels, or they could be ligand gated ion channels, right? So these are both voltage gated ion channels. Just a moment. Okay, this is voltage gated ion channels, example of that. This is a example of ligand gated ion channels. Then there could be mechanical or stress gated ion channels, and also leakage channels, which are always open, they are never closed, right? So once these ions flow through these channels, how will the concentration gradient be maintained, right? So this flow can always keep happening, right? So the two major types of ion channels, first we'll see to understand how this happens, right? One type is a voltage gated channel. So it will only be open when a certain voltage is present, right? When that voltage has gone down or it has gone up, this channel will become closed, right? So that is why it will maintain it like once it is closed, again some other thing will be functioning, and this thing will come into equilibrium, or the sodium potassium pump will be throwing these ions back out, and equilibrium will be reached again. Once it is reached again, it can open again, right? So this cycle will continue. The other is a ligand gated channel, right? This ligand gated channel, what happens? Only if the chemical molecule that is responsible for its opening is present, it will be open. If it is not present, or if it gets finished, it will close, right? So that will also be responsible for maintenance of this kind of gradient. So this effort still won't maintain the gradient. It will simply halt the flow of ions any further, right? For that, what is required? Ion pumps are required, right? That also we have already studied. They basically push ions opposed to their concentration gradient to maintain a particular charge inside and outside the membrane of the neuron. And we have seen that there is this one kind of ion pump, which is sodium potassium pump, which and which it also does this. All these ion pumps use energy to or use ATP to perform this function, and the sodium potassium pump is actually the one that uses 2/3 of the energy that is actually supplied to the neurons itself, right? So the pump consumes energy in form of ATP to maintain these gradients, and they account for 2/3 of all energy that is used by the neurons, right? So that energy, that much energy it takes, right? Now this is from here begins the second unit, right? Because we will anyway not be able to finish the second unit. I have almost covered the first unit, right? Second unit and third unit. Uh, because we are not going through the numerical part of it, so we'll quickly sift through it. So this part we'll pick up in the next session. Okay, so we'll solve it there. We'll go through it there. Yeah. So I'll just stop sharing. Yeah. It was a much less interactive session, by the way, than the previous one that we were in, right? Nonetheless, I really hope that you guys have understood the. Yeah, yes, Nibita.
In case of sagittal cord, it does not have a symmetry. How?
Sorry, can you come closer to the mic? I'm not sure if it is me who can't hear or other people also. Can you say that again?
Brain orientation, sagittal cord have not symmetry. How it is?
It will have. Because so the only one that will not have symmetry will actually be the horizontal one. You know why? Uh, because when sagittal is basically from the middle you are cutting, right? So if you're cutting into two halves from the middle, this half will also have the same organs and this half will also have the same organ, right? So it will have the symmetry. If you are doing coronally like this, right? Again, you're cutting into two halves, right? This half will also have it, this half will also have it. But when you are doing it horizontally, okay, now I understand what you are saying. So in the sagittal, yes. In the section itself, it will not be there. That is right. If we are picking one section, it will not be there. I was thinking about the whole brain. Like if we pick this half, it is similar to this half, is what I was saying. But in the section, that is not going to be visible, right? Because you are sectioning one half, right? In that, it is not symmetrical. It is not bilaterally symmetrical. It is representing one half of the brain only, right? And what I meant by this coronal and horizontal not having the that kind of symmetry was that if a coronal section is cut, for instance, then the rest of the brain was left, right? Okay, I don't want to confuse you. But that is true, right? When we are discussing solely about the sections, um, sagittal will not be bilaterally symmetrical. That is correct. Yes. Okay. So in that case, we'll pick up the week three PYQs with the week 2 and 3 discussion next Monday, right? And for now, I'll just stop the recording. Um, Thank you for attending. Yes.
Thank you,
So
ma'am.
we